Good afternoon, everyone. Thank you for having me. My name is Shani Shoham, and today I will discuss microbe-mediated molybdenum hyperaccumulation in the marine sponge Theonella conica. The work brought together marine biology, microbial ecology, electron microscopy, elemental analysis, and mineral characterization. I'm coming to this topic from a biological side. The main question I will address is simple. How can a marine animal accumulate such high levels of molybdenum, and what role might its microbial symbionts play in this process? This work was carried out in Professor Michael Lanz's lab at Tel Aviv University in a close collaboration with the material engineering group at the Technion, Professor Boaz Pokoroy's lab, whom many of you may know. First, a few words on sponges. For those of you who don't spend your time underwater, sponges are among the earliest divergent multicellular animals. They have a simple body plan, but their biology is highly complex. They lack true organs and tissues, yet they maintain strong internal organization through specialized cells, extracellular matrix, skeletal elements, and microbial symbionts. Ecologically, sponges are powerful filter feeders. They pump large volumes of seawater through their body and retain bacteria, dissolve organic matter, and particles. This constant filtration exposes them to trace elements from the surrounding water. Some sponge species can tolerate and accumulate elements at concentration far above those found in many other marine organisms. And for this reason, sponges are a useful system for studying both environmental accumulation and biologically controlled mineralization. Diving deeper into our research animal, the marine sponge Theonella conica is distributed across the Indo-Pacific region. In the Gulf of Aqaba, it is now rarely found at shallow depths, so locating and collecting fresh specimens has been a major challenge. This sponge also has a high microbial abundance. This means a large part of its biomass is composed of associated microorganisms, and one of the important symbionts in Theonella species is Entotheonella, a filamentous bacterium known for its remarkable metabolic potential. T. conica has a dark blue inner tissue. This blue color led us to suspect that molybdenum compounds might be involved. Molybdenum is a transition metal, an essential micronutrient in biology. It is used as a cofactor in enzymes involved in nitrogen, sulfur, and carbon metabolism. Examples include nitrate reductase and nitrogenase. At the same time, like other trace elements, molybdenum can become toxic when it exceeds biologically tolerated soluble concentration. In marine environments, molybdenum is usually present as molybdate. It can also occur in mineral phases such as molybdenum oxides, molybdenum sulfates, and metal molybdates. Some molybdenum compounds have blue coloration. This made the blue interior of T. conica especially intriguing. The key unknown was whether the sponge passively accumulates molybdenum from the environment or whether the accumulation is linked to biological processes inside the sponge. This led us to design a workflow that moves from the whole sponge to specific fractions, cells, and intracellular structures. We started with sponge collection from the Red Sea and compared these samples with older samples from Zanzibar. For that, we used quantitative elemental analysis to measure the concentration of 22 elements, and we separated fresh sponge material into enriched fractions. We also use SEM-EDS to localize molybdenum-rich structures in the sponge body. And to identify crystalline phase, we use X-ray diffraction. We isolated the filamentous bacteria Entotheonella for light microscopy and TEM-EDS. Finally, we analyzed the microbial community, which I will not present today. I will first show the whole sponge element data because this is where the unusual signal becomes clear. Here we measure 22 elements in sponge samples from two geographic regions, the Red Sea and Zanzibar. The most striking result was molybdenum. In the Red Sea samples, molybdenum reached an average concentration of about 46,000 micrograms per gram dry weight. And Zanzibar samples also showed high concentration of about 28,000. Despite the geographic distance, the high molybdenum signal appears in both regions. This suggests that molybdenum enrichment is likely a species-associated trait, rather than a local contamination effect. Another interesting result was selenium. Selenium was especially high in the Red Sea samples. This may be relevant because selenium and molybdenum can interact in microbial redox and detoxification processes. For this talk, the key point is that molybdenum was not a small background signal. It was one of the defined chemicals features in the sponge. The next question was where inside the sponge this molybdenum is concentrated. To address localization, we separated the sponge into enriched fractions. These include the ectosome, which is the outer layer of the sponge, an Entotheonella enriched fraction, sponge cells, and unicellular bacteria. The highest molybdenum concentration was found in two fractions, the ectosome and the Entotheonella enriched fractions. The result was important because it narrows the biological interpretation. The ectosome suggests that molybdenum-rich material may accumulate near or on the outer sponge layer. The Entotheonella fractions suggest that a specific symbiotic bacterium may also be involved. I also want to draw your attention to calcium in the ectosome. This becomes relevant in the next slide, where we look at spherical structures containing both molybdenum and calcium. After measuring elemental concentration, we move to microscopy to see where these elements appear structurally. Here, we use scanning electron microscopy with energy dispersive spectroscopy. The goal was to look for molybdenum-containing structures in the sponge body. In backscattered electron mode, as you may know, denser structures appeared brighter. This helps us screen for regions that might contain heavy elements. In the ectosome, we observed spherical granules, and EDS analysis showed that some of these granules contain molybdenum and calcium. The molybdenum to calcium atomic ratio was close to 1:1, which is consistent with calcium molybdate or powellite crystal. However, I want to be careful here. Based on semi-DS alone, we cannot definitely identify the mineral phase. We can say that these are molybdenum and calcium-bearing spherical structures and that calcium molybdate is a plausible interpretation. To test whether crystalline molybdenum-bearing minerals are present, we continued with X-ray diffraction. We use high-resolution powder X-ray diffraction to look for crystalline phases in the different sponge fraction. The analysis detected crystalline molybdenum oxide in the ectosome, Entotheonella, and sponge cells fractions. Other crystalline phases were also detected, including sodium chloride, calcium magnesium carbonate, potassium sodium chloride, and aluminium selenate. Importantly, calcium molybdate was not confirmed by XRD, and there are several possible explanations. The calcium and molybdenum-rich granules may be amorphous, poorly crystalline, present below the detection limit, or masked by the dominant salts such as NaCl. For me, the important message here is that molybdenum is present in mineral-associated forms, but the exact mineral identity is not fully resolved. Before moving to the microbial aspect of this research, I will summarize the first part of the results. At this stage, we have three main observations. First, Theonella conica accumulates high levels of molybdenum across a wide geographical range. Second, Molybdenum is enriched in a specific sponge fraction, especially the ectosome, which is the outer layer, and the Entotheonella enriched fraction. Third, microscopy revealed molybdenum-bearing spherical granules in the sponge, including structures that also contain calcium. Together, these results suggest that molybdenum accumulation is actually organized inside the sponge, and that both the outer sponge region and microbial symbionts may be involved. This brings us to the microbial community, and especially to Entotheonella, seen here on the left image. When we examined the sponge endosome by SEM, we saw a dense microbial community. This includes many unicellular bacteria, seen here on the right, and a high abundance of filamentous bacteria identified morphologically as Entotheonella and later also genetically. This was important because Theonella swinhoei, a closely related sponge, is also known to accumulate metals such as arsenic and barium through processes associated with a similar symbiont, Entotheonella. Therefore, we ask whether the microbial community of Theonella conica differs from Theonella swinhoei and whether Entotheonella might also be connected to molybdenum accumulation. To make the story short, it's not the same bacteria, but it's from the same genus. And during the isolation of Entotheonella from fresh T. conica tissue, we observed striking intracellular spheres under light microscopy. Many of these spheres had blue color, and some also showed yellowish regions. Because molybdenum compounds can show different colors depending on their oxidation state and chemical composition, this observation immediately suggests that these intracellular structures might contain molybdenum. At this point, the observation was only visual, and it was exciting but not sufficient. We therefore prepared the bacteria for TEM-EDS to test whether molybdenum was actually presented inside these structures. Here we examine thin section of isolated Entotheonella cells using TEM combined with EDS. Inside the bacterial cells, we observed high-density spherical structures. EDS confirmed that these spheres contain molybdenum. Across the analyzed spheres, molybdenum was measured at about 14.2 atomic percent and about 13.3 mass percent. Other dominant elements included oxygen, carbon, calcium, sulfur, and iron. And this is the central result of the study. Entotheonella cells contain intracellular molybdenum-rich spheres. The FFT analysis, however, did not detect a clear crystalline signal from these spheres. This suggests that the material may be amorphous, disordered, or mixed phase that is difficult to resolve by this method. I will phrase this carefully. These results support intracellular molybdenum accumulation by Entotheonella, most likely through a biologically induced mineralization process. To connect this intracellular evidence with the bulk mineral data, we return to XRD. The XRD analysis detects molybdenum oxide in several fractions, including the Entotheonella fraction. This supports the presence of molybdenum-bearing crystalline material in the bacterium enriched fraction. However, the intracellular spheres observed by TEM did not produce a clear crystallographic signal. Therefore, XRD and TEM-EDS should be interpreted together. XRD tells us that the crystalline molybdenum oxide exists in the fraction, and TEM-EDS tells us that the molybdenum-rich spheres exist inside Entotheonella cells. The exact phase inside intracellular spheres remain unresolved. For a material science audience, this is an important open point. Future work should combine higher resolution structure analysis, oxidation state analysis, and targeted isolation of these spheres. I will now summarize the biological interpretation. The combined evidence suggests that Entotheonella contributes to molybdenum mineralization or sequestration in Theonella conica. The strong evidence comes from the intercellular molybdenum-rich spheres observed by TEM-EDS. Calcium, sulfur, and iron were also present in these spheres, suggesting that these elements may participate in the mineralization process, and the lack of clear crystalline signal from the intercellular spheres suggests that the material may be disordered or amorphous. This resembled previous finding in Theonella swinhoei, where Entotheonella was associated with intracellular mineralization of arsenic and barium. I will not claim that we have resolved the full mechanism. Instead, I will say that our data support a model in which Entotheonella acts as a microbial compartment for molybdenum accumulation inside a sponge. This study was published in Science Advances journal in July 2024. For me, the significance of this work is that it connects three fields: marine symbiosis, microbial biomineralization, and the formation of metal-rich biological microstructures. It also shows that marine sponges can serve as a natural system for discovering unusual metal accumulation and mineralization processes. One final note before I finish. A recent preprint by Dr. Tutopalli and colleagues reported a spontaneous formation of molybdenum-rich protocell-like compartments. They use a simple aqueous mixture containing formaldehyde and inorganic salts, including molybdate, and under the experimental condition, the mixture formed blue spherical compartments. These compartments showed growth, internal structure, and internal seating. The author compares their synthetic compartments with the molybdenum-rich structure we observe inside Entotheonella, and this preprint does not prove that the same mechanism occurs inside a sponge, but for material science audience, this is a broader point. The natural system may offer clues of how living cells control metal-rich microcompartments, while synthetic system may help us test which part of the process can occur without biology. I would like to thank my collaborators from the Faculty of Material Engineering at the Technion, especially Professor Boaz Pokoroy and his team. I also thank Professor Michailan and my colleagues from Tel Aviv University. This research was founded by the Israel Science Foundation. Thank you for your attention. I will be happy to take questions, and you are welcome to write to me. My current position is at the Kinneret Limnological Institute. Thank you.